Good morning, good afternoon, and good evening, everyone. And welcome to the Scientist.com and Stem Cell Technologies webinar titled Implementing the Colony Forming Unit Assay as a Potency Assay for Hematopoietic Cell Therapy Products. I'm Liam Sanya from the events team here at Scientist.com, and I'll be your host today. We're very fortunate to be joined by Dr. Colin Hammond, scientist in research and development at Stem Cell Technologies. Today, Colin will discuss some of the regulatory guidance around hematopoietic cell therapy products and how to validate the CFU assay as a potency assay. And with that, I'm very pleased to welcome Dr. Colin Hammond. Colin, thanks so much for joining us today, and the floor is yours whenever you're ready.
Great. Thank you, Liam. Hello, everyone. Thank you for taking your time to watch this webinar. I'm Colin Hammond, a scientist with Stem Cell Technologies. As a disclosure, I'm not a clinician or directly involved in the manufacture of cell and gene therapies or their regulatory status. My expertise is in the biology as well as the functional and molecular assessments of human hematopoietic stem and progenitor cells. And today I'll be discussing methods and considerations for using the colony forming unit assay, more commonly known as the CFU assay or CFC assay, as a potency assay for hematopoietic cell therapy products. The objectives of today's webinar are to understand the role of bioassays as a potency assessment in hematopoietic cell therapy workflows, and the bioassay we'll focus on is the CFU assay. So we will discuss what it is and how we use it as a measure of hematopoietic stem and progenitor cell functionality. And we will go through strategies and considerations to validate the CFU assay in-house, as well as techniques to improve the throughput and standardization of the assay. A brief outline of the rest of this talk. We'll start with what some of the current regulatory guidance is surrounding hematopoietic cell therapy products, and take a look into the biology of hematopoietic stem and progenitor cells, which I will frequently refer to as HSPCs, and the methods that we use to evaluate them. And of course, the use of the CFU assay specifically in evaluating potency with a focus on the validation and utility of reference standards for the assay. So what are HSPCs being used for? Well, hematopoietic stem cell transplantation, or sometimes more simply called hematopoietic cell transplantation, has been used since the 1970s to treat a variety of cancers and other diseases in patients. These transplants formerly relied on cells harvested from the bone marrow, but advances in HSPC mobilization strategies and the use of cord blood units has increased the availability of donor cells suitable for transplantation, with well over 50,000 transplants being performed annually. In addition to the transplantation of unmanipulated or minimally manipulated cells, the transplantation of genetically modified HSPCs in the treatment of several diseases has been increasing, with over 400 patients having received genetically modified HSPCs as of 2022. Several hereditary disorders that are potentially curable by gene editing of HSPCs are shown here. Importantly, each product is unique, and there is a large amount of variation in the gene transfer strategy and HSPC transplant dosage being used in the clinic. Currently, the FDA has approved 12 hematopoietic cell and gene therapy products, some minimally manipulated cells, and others using expanded or gene edited cells. Since these products are ultimately intended for use in patients, there are regulatory requirements that must be met. Different regulatory agencies have released guidelines and recommendations to help direct the developmental process and ensure that you familiarize yourself with these guidelines early on to avoid any unnecessary delays. For today, I'll mainly be highlighting recommendations from the FDA, but the regulatory requirements for other authorities, such as Health Canada, have similar requirements for the assessment of product quality, biopurity, and potency testing for cell therapy products. And of course, all steps for approval of a cell and gene therapy product should be pursued with close contact with the relevant regulatory authority. So for products relying on the use of primary human samples, the variability of the starting material can make it difficult to maintain reproducible lot-to-lot consistency. The requirements for assuring the quality of a product is outlined in the FDA guidelines, stating that no lot of any licensed product shall be released by the manufacturer prior to completion of tests for potency, sterility, purity, and identity. So to assess that the product is of high quality, measurements including evaluation of the purity and potency of the product need to be done at various stages of the manufacturing process. Measuring the purity of a product requires characterization of the cells of interest in the product, as well as the level of any contaminating cells. And the potency of a product is more difficult to define since the effectiveness of different products may rely on different factors. Therefore, the FDA definition of potency is a measurement of the capacity of the product to affect a given result, and tests for which are expected to correlate with the intended clinical response. And an important note here is that a measure of viability of the cells in a product, while important and necessary, is typically not acceptable as the only indicator of functional activity. And we'll go into this in more detail later on as we discuss the differences between purity and potency assessments. A part of these recommendations is to develop potency assays in the early stages of product and process development, which will help guide the overall manufacturing process. Furthermore, it is suggested that the early phase of development is actually accompanied by trying several different assays comprising a comprehensive testing matrix to see what works within the manufacturing workflow and what actually correlates well with the preclinical and clinical data as it is collected. Progress through the different stages of development may change aspects of how a potency assay is performed, but it will eventually require standardization to a level of assessing whether a product meets specifications and is therefore suitable for release. These target profiles and pass criteria for a given product are determined in connection with the data generated during the development process and will likely be specific to a given product. Okay, so we've talked about the requirement of potency assay incorporation into product development workflows, but how do we actually measure the potency of hematopoietic stem and progenitor cells in vitro? Well, the gold standard for this is the colony forming unit assay, or the CFU assay, due to its decades of use and demonstrated high correlation with transplant outcomes. And in this section, I'll explain some of the basic biology of these cells and features of the CFU assay that has made it such a useful assay for HSPCs. As a refresher for everyone, the blood system is highly cellular and it's constantly being refreshed. The entirety of this system is collectively maintained by a small fraction of cells called hematopoietic stem cells, or HSCs. And in humans, there are estimated to be a few thousand of these in a person. And they actually divide very infrequently, at most a few times a year. But nevertheless, they produce an estimated 140 trillion, with a T, mature cells over the course of a year. To put that into a little bit more context, the total square footage of Canada, where Stem Cell Technologies is headquarters, here in Vancouver, British Columbia specifically, is only about 110 trillion square feet large. So, how do so few cells produce so many? The answer, of course, is through a series of intermediate progenitors, which have differences in their functional capabilities as we move from progenitors relatively close to the HSC portion of the hierarchy to progenitors that are closer to the terminally differentiated mature cell fraction. Taking a closer look at the hematopoietic hierarchy, we can see many subsets of cells. In this diagram, we can see the stem cell pool represented on the left and morphologically distinct mature cells on the right, with the progenitor intermediates bridging the two sides. In general, as you move from left to right in the diagram, the cells lose the functionality of stem cells, that is, namely their ability to self-renew. As well, there is a general restriction in the lineages that a cell is able to produce, a move from being able to produce all lineages of blood cells to only being able to produce cells of a single lineage, such as lymphoid or myeloid. In practice, the majority of human hematopoietic stem and progenitor cells can be identified using the CD34 surface marker phenotype. Functional testing of cells within this hierarchy requires the use of specialized assays that are able to allow a cell to exert its functionality. For example, the gold standard for showing you have definitive functional hematopoietic stem cells is through showing the production of human cells over a long period of time, generally 4 to 6 months, after transplanting into an immunodeficient mouse strain such as the NSG. Moving to in vitro systems, the long-term culture initiating cell assay detects cells generally a little downstream from the true HSCs using a specialized co-culture system and stem cells mylo-called H5100 medium. Assessment of progenitors' lymphoid potential has historically also required the use of co-culture systems to support the growth and differentiation of identifiable lymphoid cells, such as using the MS5 stromal cell line for B cell generation or the OP9 DL1 or DL4 line for T cells. However, stem cell now also offers stroma-free systems for particular lymphoid cell production, as shown here. The myeloid and erythroid potential of HSPCs can be quantified in vitro using the colony forming unit assay or CFU assay. And very importantly, performance in the CFU assay has been shown to be highly correlated with transplant outcomes in patients arising from decades of clinical data. And Stem Cell provides a comprehensive portfolio of products and services to support the use of the CFU assay. Okay, so going into more detail about the CFU assay, versions started in the mid 1960s with the aim of ultimately trying to measure the number of functionally viable HSPCs in a population. I mentioned earlier that the CFU assay probes the ability of cells to produce myeloid and or erythroid colonies, and a cell that is capable of producing both, as well as megakaryocytes, is termed a colony-forming unit, granulocyte, erythrocyte, monocyte, megakaryocyte, or more simply as a CFU GEMM. Those with myeloid-only colonies are either CFU-GM, if containing both granulocytes and monocytes, or just as CFU-G or CFU-M, if only one or the other. And those that produce colonies with only erythroid cells are termed blast-forming units erythroid or colony-forming units erythroid, depending on their size. Progenitors with megakaryocyte only potential are generally difficult to discern in the standard CFU assay, but instead can be examined using Stem Cell's megacult system, but I won't really be going through this anymore today. On the right-hand side, we can see representative images of some of these different colony types, with the erythroid cells having a distinct reddy brown color due to their iron-containing hemoglobin. Plating and culture of the cells in a semi-solid medium, such as our methicalled medium, is a very important feature of this assay since it restricts the movement of a cell and allows for growth to be attributable to an individual clone. This means the CFU assay is a quantitative assay, that is, when it's performed properly, a colony will originate from a single cell. So a colony forming unit, or a CFU, refers to a single progenitor that gives rise to a single colony in the assay. It follows then that more colonies observed in the CFU assay indicates that there were more viable progenitors in the initial population that was plated. And if there are fewer colonies seen, then that means there are fewer viable progenitors in the originally plated population. So in general, how is a CFU assay performed? Well, it can be broken down into 4 steps. The first is preparing your cell suspension, then adding these to your methicalled medium, dispensing that medium into plates or dishes, and incubating them, and then finally scoring your plates by enumerating the colonies. And here we can see some representative images of CFU plates that were initiated with increasing numbers of CD34 positive cord blood cells. And as I mentioned, if you plate more functional progenitors, you will end up with more colonies up to a point of saturation. Those looking for training on performing all steps of the CFU assay can take the Stem Cell On-Demand training course or find additional tips and how-tos on our website. Due to the utility of the CFU assay in evaluating the potency of HSPCs, it is being actively used in the clinic. Among currently approved hematopoietic cell and gene therapy products, all of them reference the CFU assay. And here you can see a breakdown of where in approved product workflows the CFU assay is mentioned. Older products refer to the CFU assay as part of product preparation as per transplant center procedures, whereas newer products such as Casgevy and Zynteglo typically recommend CFU counts as part of release specifications and quality control. Speaking of quality control, another important testing area for cell and gene therapy products is in stability programs. And stability testing is a key part of product quality assurance, indicating that a product continues to meet its criteria after the influence of environmental factors or storage conditions. The FDA recommends the inclusion of potency assays as part of a stability program and indicates the use of colony-forming assays may be a part of such a program. The FACT-JC guidelines point to the testing of cryopreserved cell therapy products for viability and potency on samples representative of the processing methods and storage duration of clinical samples. AABB similarly outlines potency as a required testing metric with a relevant assay left to be defined by the facility. And I've also included here the soon to be published draft of the NetCord FACT guidelines requesting potency be demonstrated by the CFU assay or other validated potency assay prior to sample release. So now that we know what the CFU assay is, and that it is being actively used in the clinic and recommended by some of the regulatory guidelines, we'll go over how and why product purity and potency are not interchangeable and how potency assessments using the CFU assay can capture product quality failures that can be missed by using purity assessments alone. And for this, I'll focus on a particular use case of the CFU assay in stability programs, such as the case for cryopreserved products. In the majority of the regulatory guidelines, the specifics of potency assays have been left intentionally vague. So why is a measure of viable CD34 positive cells, that is a measurement of the number of viable HSPCs in a product, typically not sufficient to indicate the potency? While we know that dose thresholds for many transplant centers are based upon the number of CD34 positive cells, and using methods such as the ISAGE protocol of flow cytometry assessment, we can standardize how we measure this number and can generally see very good positive correlation of transplant outcomes with the number of viable CD34 positive cells transplanted. Well, it is important to recognize that the CD34 positive cell compartment is actually highly heterogeneous, as you saw earlier in the hierarchy figure that I showed. And extending this, not every CD34 positive cell is functionally potent. For example, typically less than half of CD34 positive cells are measurable as CFU. More often, this is in the range of 10 to 30%. And only 1% or so of CD34 positive cells are bona fide HSCs capable of long-term engraftment. However, reports that have integrated use of the CFU assay to functionally interrogate the CD34 positive cells in a sample have shown good positive correlation with a number of CFU GMs, that is the number of progenitors which form colonies containing granulocytes and monocytes, with engraftment outcomes such as time to neutrophil and platelet recovery in patients. As well, in cases where insufficient numbers of CD34 positive cells were present in a sample to be transplanted, as long as the CFU GM numbers exceeded a certain threshold, they were actually performed quite well in transplants. And I do want to highlight here that most of the time, the number of CD34 positive cells in a sample and its performance in the CFU assay are very well correlated. And this is expected since the CFU assay was developed to measure these viable progenitors. However, in certain circumstances, this correlation falls apart, showing the insufficiency of relying purely on CD34 cell numbers as an indicator of sample quality. So as an example of this, we can look to a scenario that occurred years ago at the Great Ormond Street Hospital in the UK, where transplants being given to children were being unexpectedly ineffective. To address this problem, samples were sent for analysis by Dr. Watts and were found to have good cell numbers and viability, same as had been seen using the hospital protocols. But when Dr. Watts performed an additional test with the CFU assay, he found the cells were unable to grow properly, echoing the inadequate potency of the cells that were transplanted. The cause of this was eventually traced to errors during the freezing of the samples, and his recommendation was then to incorporate the use of the CFU assay as a method of validating the freezing process of samples for transplantation. But how could the number of viable CD34 positive cells be misleading as to their potency? Well, it's important to note that the majority of dye-based viability assessments actually infer viability from the membrane integrity of a cell. That is, when cells are viable, their membrane is intact, and non-permeable dyes are unable to enter. When a cell is dead, or at least quite far into the apoptosis process, Their membrane is disrupted, and the dye can enter and stain interior components of the cell. And when we use these types of viability methods for fresh cells or those from cultures, the correlation is generally quite good. However, when we're using this methodology after cryopreservation, what we are observing is essentially the frequency of cells that have maintained an intact membrane throughout the freeze and thaw process. However, cell stress response pathways can be activated during this process, which result in apoptosis or necrosis hours post-thaw, as shown in the figure on the right. So performing only a post-thaw viability assessment underestimates the proportion of cells that display this delayed onset death, and moreover, longer-term functional impacts are not captured by viability assessments alone. If we return to the real-world example from the Great Ormond Street Hospital, an investigation from Morgenstern et al. Found that a suboptimal freezing rate was responsible for the lost functionality of their cryopreserved cells. This criticality of freezing rate was also examined in a paper from Yang et al., which demonstrated that as cells are transferred at too warm of a temperature to a liquid nitrogen freezer, then you observe a disconnect in the number of viable CD34 positive cells you recover post-thaw and the actual ability of those cells to form colonies in vitro. That is, the cell's potency, therefore, could not be anticipated from a post-thaw viability assessment alone. We performed a similar experiment in-house to recapitulate this observation, and similar incorporation of a potency assessment post-cryopreservation is actually recommended by these regulatory agencies, which I've mentioned. So here we used 3 cord blood donors enriched for CD34 positive cells and froze them in 3 different ways. For the first being a normal freezing using 90% FPS and 10% DMSO with a Mr. Frosty passive freezing device, which cools at 1 degree per minute into a -80 freezer overnight before transferring to a liquid nitrogen freezer the next day. A condition with the same cryoprotectant medium, but this time transferring ice cold samples around 4 degrees directly to the liquid nitrogen freezer. And lastly, as a negative control, cells that were frozen with the normal procedure, but without the addition of DMSO as a cryoprotectant. After thawing, we assessed the viable CD34 positive cells we were able to recover from each sample and performed CFU assays to gauge their potency. Very importantly, we ran each CFU assay alongside a pre-validated reference standard, and I'll go over why this is used and what it can show over the next few slides. Okay, so as you can see in the figure below, the recovery of the normal frozen samples is around 70%, with the suboptimally frozen samples being more variable but maintaining relatively decent numbers of viable cells post-thaw. Those without DMSO, unsurprisingly, do quite poorly. However, when we look at the results of the CFU assays, we find that only the normally frozen samples of any colony forming ability, and therefore any potency remaining after cryopreservation. You can see this striking result from these representative images from the CFU assays on the right, showing colonies only in the fresh and normally cryopreserved samples with very few to none in the suboptimal and DMSO-free conditions. Now, interestingly, we had actually performed this experiment multiple times. And here are 2 examples that shared the same design and reagents, but were performed on different days. The CFU assays were actually cultured in different incubators as well. And this is what the data looked like for these 2 experiments. In experiment 1, all conditions showed low numbers of colonies in the CFU assay, and in the second experiment, the numbers were much higher. Now, on the previous slide, I mentioned that we set these experiments up alongside a pre-validated reference standard. And during validation of this reference standard, we determined the experimental range that the standard has, as shown here. When we compare the results of the reference standard in our 2 experiments with the expected range, we see that the results of experiment 1 are well below our expected range for the reference standard, indicating that some error occurred during the experiment. In this case, it turned out to be a malfunctioning incubator. But without the reference standard being run alongside for each of these experiments, we would be unable to determine whether the results of experiment 1 were valid or not for the test samples being examined. So importantly, one use of a reference standard is to indicate if there is an underperformance in the reference standard, usually means an error occurred in the experiment. So in this next section, I will give an overview of how to validate a potency assay, in this case, the CFU assay, and go over the validation and utility of reference standards that I just mentioned for the CFU assay as a tool for standardizing your testing workflows and confirming the validity of experimental data. If we return to the FDA guidelines for industry, I've pulled a few key points out of their recommendations. Firstly, the potency of biological products should be measured quantitatively in a way related to its ability to affect results. That is the FDA definition of potency. And this can be done in vitro or in vivo with specifically designed methods to satisfactorily meet the FDA definition of potency. Importantly, they further suggest the use of reference material to ensure the assay performance is as expected, and that this reference material should be developed in-house, meaning that it needs to be validated. So those latter 2 points refer to using a reference material as an internal comparison for the potency assay. This is using a relative potency approach where results of test samples are not reported absolutely, but instead as a percent of control. In the case of the CFU assay, a reference standard is a sample validated for performance and consistency in the assay, and this can help to combat inherent donor-to-donor variability, can be used to establish the accuracy and robustness of the assay, aids in the evaluation of critical reagents prior to use, and for bridging of changes to the methodology between new lots and old lots of reference standards as well. And from a quality control perspective, enables the review of trends over time, such as through the use of control charts. And the image on the right outlines that a potency assay, as I mentioned earlier, should be incorporated quite early into the product development life cycle, as it can give insights for later stages and in terms of determining acceptable thresholds for your product. But also that this potency assay may need to be tweaked at several different steps as processes and materials are finalized all the way through approval and the final release stages. So what are some considerations for validating the CFU assay as a potency assay? The first, of course, is determining if it is feasible and a proper assessment of your product's potency. If you are wanting to assess the functionality of engineered CAR T cells, the CFU assay is likely inappropriate. However, if you were to look at the potency of HSPCs to be used to generate CAR T cells in a differentiation strategy, it therefore might be appropriate. A validated potency assay requires, as appropriate, measures of specificity, linearity, accuracy, precision, range, and detection limits. And to address these parameters, the rule of 3 is recommended. That is performing 3 replicates of each test concentration with at least 3 test concentrations, repeated across 3 independent experiments. In a CFU assay, following these guidelines, we would have 3 test concentrations at least. We would add enough of each cell suspension to enough methicult to plate across 3 wells or dishes, and we would repeat this at least 3 times. So as an example, this is what we did recently. Here, previously frozen cord blood CD34 positive cells were plated in triplicate across 4 cell concentrations in 3 replicate experiments. And again, we are plating these multiple cell densities to identify the linear range and accuracy of the assay, as well as the detection limits and precision. In the figure on the right, we can see the plotted results of these experiments with the number of colonies in the CFU assay on the y-axis and the number of plated CD34 positive cells on the x-axis. And each individual line represents an independent experiment. From the chart, we can see the response is linear over the range of cells tested. The raw data from one of these replicate experiments is also shown in the table below, and you can see on the right-hand side of the table a calculation for the CV of the results. So what is that? The CV, or coefficient of variation, is a reflection of the standard deviation of a set of observations divided by their mean, and of course multiplied by 100 to get a percentage. The CV is a useful metric for evaluation of other assay parameters. For example, calculation of the intra-assay CV evaluates how precise your replicates are and the accuracy of your results across multiple concentrations. That is, how disparate are the results extrapolated from different plating densities. Inter-assay CV evaluates how robust your assay is. Shown in the table below is a summary of the data from 3 replicate experiments and the calculation of the inter-assay CV from the same. As a general guideline for the CFU assay, we suggest that the intra and inter-assay CVs should be below 20% when using the same conditions, reagents, and protocols. So what does the overall workflow of validating a reference standard look like for the CFU assay? For a cell-based assay, you will need to have cells. So starting with a primary sample, and this could be from a single donor or from a pool of multiple donors, which can be very useful to have more material to work with. You will thaw and count your viable cells. I say thaw here because you typically want to be able to return to a reference standard quite frequently. So long-term storage is a must. And then perform your purity analysis, for example, an assessment of CD34 positive cell content. You will then need to prepare your cell suspensions at multiple densities. We suggest making your suspensions at 10 times the desired plating density, for example, a 5,000 cell per milliliter suspension for a 500 cell per dish plating density. There will be cell suspensions prepared across at least 3 densities. And for initial assay feasibility and development, performing more than 3 densities could actually help in establishing the linear range of the assay for your reference sample, which can then be dropped to fewer densities once the range is established. The cell suspensions will then be added to a sufficient amount of methicult to allow for plating in triplicate. That would be 400 microliters of cell suspension to 4 milliliters of methicult. These are then plated in 1.1 milliliter volumes into separate 35 millimeter dishes or individual wells of 6-well smart dishes. And once plated, you would incubate your cultures for 14 days to be able to distinguish colonies by morphology. I will mention, however, that for users interested more in total colony numbers, as opposed to specific phenotypes of those colonies, we do have a methicult formulation allowing for 7-day incubation, and I'll show an example of that a little later on in the presentation. After incubation, colonies can be scored manually or with automated methods, and these counts are used to calculate your assay validation statistics, similar to what I demonstrated previously. And this will be performed at least 3 times to validate your reference standard. And when moving to examination of test samples, the workflow will be largely the same, but will require running of the test samples in parallel to the reference standard and reporting the results as relative, following the relative potency strategy I discussed earlier. Okay, now that we have gone through how and why to perform a CFU assay and how to validate a reference standard, the next question you may have is, where does the CFU assay fit into our workflow? To help answer this, we've generated a toy workflow for a gene editing of CD34 positive cells with some example points of where a CFU assay can fit and what types of information the result of that assay will give. So starting with a primary sample, this could involve isolation of CD34 positive cells or other initial sample processing steps. On this processed sample, a quality analysis, including things such as cell count, viability, purity, and CFU assay performance, would give an indication of the quality of the processed unit. And let you ask the questions, is this unit suitable for storage or other downstream steps? Did our processing methodology compromise the quality of the sample? Afterwards, if there is a cryopreservation step following processing, then a post-thaw quality assessment is important to understand how the freezing and thawing process impacted the sample. Here, performing a CFU assay can help to discover quality defects that CD34 positive viability is unable to do so, as we went through earlier. Actual editing strategies will, of course, be method specific, but in general, a quality assessment on the cells following gene editing protocol will give an indication of how the methodology of gene transfer itself impacts the quality of the cells. Some workflows may include a post-gene editing recovery period that would also present an opportunity to assess the functionality of the modified cells after this period. And just a note here as well, since the colonies in the CFU assay arise from individual HSPCs, if you were to pluck these colonies and assess them for editing, it would give an indication of the editing efficiency in your actual functional HSPC population. So what does the CFU assay look like at each of these quality assessment points? Since FDA guidelines suggest running potency assays alongside a validated reference standard, we will set up in parallel your reference standard and test samples, plating each in triplicate at 3 different concentrations. After incubation and scoring, the next step is to determine if the performance of your reference standard is in line with your validation data. To achieve consistent performance of the reference standard, the assay setup and scoring requires standardization. If your reference standard performance passes, then the experiment has gone well, and the results of the test samples can be interpreted as a relative potency to the results of the reference standard. If the performance of the reference standard fails, then these are likely reflecting technical or experimental issues in the experiment, such as changes to the method, issues with a reagent or storage, inconsistency with scoring, and this could include things like equipment malfunctions, as I highlighted earlier. It is also important to note that when bridging new lots of reference standards or materials, this would sort of be the general workflow to follow as well. Okay, on to the last section of the talk. How can we standardize and streamline the CFU assay so we can get unbiased and consistent results? Well, it is important to recognize that the CFU assay is a bioassay, and like any biological system, it's therefore prone to variability. So implementing standards around key parameters can reduce these sources of variability and help to ensure consistent results. Regulatory recommendations to control variability include the use of qualified reagents with long-term availability, such as Methocult medium. Standardization of practices are also recommended by implementing standard operating procedures and ensuring adequate staff training. And last but not least is standardization of what is most subjective part of the CFU assay, which is the counting and identification of the colonies. This can be done with rigorous training of manual operators, but automation is recommended as a method to reduce inter-operator bias and to improve consistency while also accelerating the data analysis. The CFU assay requires counting and identifying the colonies present in each assay, and this has a level of subjectivity to it because colonies can overlap or have difficult to discern morphologies. This subjectivity can introduce bias and variability in the results. Reducing this variability for human operators requires proper training and refreshers. Part of this can be done through enrollment and proficiency testing programs, which evaluate all steps of CFU assay performance, or using an automated scoring method to streamline the workflow, and as I mentioned, help reduce this operator-to-operator bias. And you can see an image of our StemVision instrument here on the right, as well as an example CFU image, which has had its colonies automatically counted and classified by cell type by the StemVision instrument's analyzer software. The identified colonies are circled with colors distinguishing colonies of different types, and the automated counting has been validated for use with normal healthy samples of cord blood, bone marrow, and mobilized peripheral blood samples. External validation of the StemVision instrument has also been reported in a study published by Vellier et al., which found the StemVision platform complies with the regulatory guidelines for repeatability, interoperator variability, as well as intermediate precision and method comparison. Importantly, like all methods in a CGT workflow, use of automated scoring requires internal validation for your particular use case. As an example of evaluating this, using pre-validated reference standards may be a good source to investigate potentially advantageous changes to the analytical methods in your workflows. Here, we use validated reference standard samples in Methocult across 4 plating densities in triplicate, following the rule of 3. We also use 3 separate lots of Methocult for this experiment. colonies were scored manually and with the STEM Vision Instruments automated counting software, and results for each plot are the average of 3 independent experiments. So what we can see from these results is a very high correlation between the number of colonies identified by manual scoring in the solid lines and the number of colonies identified by the STEM Vision software in the dashed lines. And if you summarize these results as cloning efficiency, as in what percentage of plated cells produced a colony, the results are not significantly different. And we would conclude that the STEM Vision counts are highly correlated with those of a manual operator in this scenario. In a similar vein of increasing throughput, the standard CFU assay requires 14 days to allow colonies to grow and become morphologically distinguishable, such as allowing erythroid colonies time to hemoglobinize and become noticeably red in color. A modified version of our Methicol medium allows for readouts of total colonies in about half the time. In general, when you're looking to make a change to the method in your workflow, you require bridging between the methods to show that the change is as good or better with an added advantage by way of information, turnaround time, or cost. And this requires showing data on your old method and match data using the new method. Here, for example, is a set of data showing how validated reference standards set up in both a normal 14-day CFU assay and in our 7-day CFU assay using Methicol to express, following the rule of 3. And what you can see here is there are no significant differences in colony formation assessed either at day 7 or day 14 between these two different setups. And of course, as with everything else I've discussed today, internal validation must be performed for your specific products and workflows and should follow recommendations by the relevant regulatory agency. With that, a summary of what we discussed today is that the manufacture of cell and gene therapy products requires assessment of several product quality parameters at various stages of the workflow, including potency. Regulatory agencies have many guidelines for what potency assays should be able to do. The first being it should measure product activity. Due to extensive historical research supporting the correlation of performance in the CFU assay, which HSPC functionality and transplant outcomes, we can also use the CFU assay to reveal functional issues in HSPC products that are not revealed by purity assessments alone. Potency assays are also required to have validated parameters. This requires them to be quantitative, and the quantitative results of the CFU assay allow for validation of parameters such as linear range, accuracy, precision, and robustness as required. Also, our recommendations for the use and internal validation of reference standards, and these can aid in method optimization and ensure consistency over long development periods, and can be also useful for bridging between methodologies, validating new reagents, identifying trends in your results and processes. In terms of standardization of the CFU assay performance is fairly sensitive to reagents and culture conditions. Using high quality and stable products can help to standardize this assay. Switching from manual counting methods to automated counting methodologies, such as using the STEM Vision instrument, can also help to streamline workflows and reduce variability. And lastly, for those that are currently using or considering the CFU assay as part of their workflow, know that StemCell provides a comprehensive portfolio of reagents and services to support the use of the CFU assay in any workflow from basic research, toxicity testing, or drug testing to clinical applications. We also offer primary cells, qualified mediums, personalized courses, as well as QC kits and clinical support to help guide use of the CFU assay implementation and standardization in your workflow. We also offer contract research opportunities for clients wishing for this workflow to be performed externally. With that, I would like to thank everyone for listening to this presentation, and I am more than happy to take any questions. Thank you.
Fantastic. Well, thank you, Colin, for a great presentation. We'll now move on to the Q&A session. And as a reminder, if you do have any questions that you'd like to ask, you can submit it using the Q&A tab on the right-hand side of your screen. All right, Colin, we'll start off with a good question here. Do cells from cord blood in adults behave differently in the CFU assay? Oh, sorry, I think you might be muted.
Oh, perfect. There we are.
There you go.
Hear me now? Excellent. Yeah, cells from cord blood and adults do behave slightly differently in the CFU assay. Cord blood cells are generally more proliferative than those from adult sources. So you'll see, for instance, the size of the overall colonies will be quite a bit larger from cord blood than they will be from mobilized peripheral blood or from adult bone marrow. You can also see some differences depending on exactly what sources you're comparing in terms of the frequency of myeloid and erythroid colonies from those different sources as well.
Awesome. There's another sort of question along the same vein here. They asked what cells you use for the CFU assay. You're talking about cord blood cells, but could bone marrow cells be used as well?
Yes, absolutely. So the CFU assay has typically been set up for use from normal, healthy samples of adult bone marrow, mobilized peripheral blood, and cord blood, though it does have uses beyond that as well. The important thing is that it will need to be validated for your workflows, for your cell types of interest. If this includes genetic modification of your cells of interest prior to placing into a CFU assay, of course, you'll want to validate that this has some correlation with your desired clinical outcome. Same thing if it's an expansion protocol to try to expand your CD34 positive HSPCs or any other modifications that you might be doing.
Excellent. So talking about modifications, can you use this CFU assay for expanded or genetically modified CD34 positive cells?
Absolutely. So you will notice differences between unmanipulated cells plated in the CFU assay from those that have spent some time in culture. Usually longer periods of time in culture will have a greater effect on how they will perform in the CFU assay. Again, depending on your purpose and your desired clinical outcomes, those correlations will need to be made for the performance of your product at the time of testing with eventual clinical responses.
Excellent. Yeah, sounds great. Another question here. Can one sort single cells into plate wells with the methicult media to try to make sure that the final colonies come from that specific sorted cell?
Yeah, fantastic question, and one very close to me. I co-authored a publication on this several years ago where we did exactly that. We performed index sorting of single cells across the CD34 positive hierarchy, plated into our methicult medium. So every colony that we had come out, again, arose from a known original single cell that we could trace back to a specific phenotype and specific cell progenitor within the CD34 hierarchy. That was all using cord blood. For that, there was no clinical correlates that we were trying to do. This was purely a biological academic exercise. So if that is something that is desired to be used as a potency assay for something more clinical, again, those correlates would need to be determined and validated.
All right, very cool. Nice. All right, next one. Somebody says, I evaluate cytotoxicity of CAR T cells on stem progenitor cells. In every group, I talk to use cord blood cells for the CFU assay when evaluating the CAR T cells. Do you think it's worth using CD3 positive cord blood cells or bone marrow cells from an FDA point of view?
Yeah, so this is a great question, and I think I touched on it a little bit during the presentation, where the CFU assay is really set up to evaluate the functionality of progenitors in your starting sample. So if you have already taken your cells through a differentiation process where you have depleted a lot of the progenitor activity, after that point, the CFU assay may not be appropriate for your specific workflow. If you were looking more upstream for donor screening or initial cell processing and storage of progenitor units to eventually be used for this differentiation strategy, then the CFU assay would be very appropriate. Once you have already differentiated them or manufactured them into CAR T cells, a more T cell specific functional assay should be used.
Excellent. Okay, so since only 10 to 30% of CD34 positive cells can be measured as CFU, does that mean that the rest of the cells are not early progenitors, or the rest of the cells have very poor, stemness that can't be measured? And does this mean that only 30% of the 30 CD34 positive cells maintain potency generally?
Yeah, so this is a really fantastic and biologically complex question that I'll try to answer um, in a short period of time. So, of course, it's going to gloss over some of the specifics. 10 to 30% of CD34 positive cells being measurable as CFU, sort of on average there. Again, the CFU really reads out cells that have erythroid and myeloid potentials. There is also a size threshold for cells needing to complete certain numbers of divisions so they can make an identifiable colony. Cells that are more downstream in the CD34 compartment may not have that amount of proliferation in them to be able to perform make identifiable colonies in the CFU assay. Those that have only lymphoid potential, for example, as well, would not be read out efficiently in the CFU assay. And typically highly primitive, more towards the HSC compartment, typically don't read out well in the CFU assay, mostly as a measure of time. The CFU assay is a 2-week assay for reading out very highly primitive cells. As I alluded to in the presentation, the gold standard still remains in vivo transplantations, or you can use an in vitro long-term culture initiating cell assay, which extends the in vitro culture period up to 8 weeks or so. 6 weeks in a co-culture, followed by harvesting, and 2 weeks in a CFU assay.
All right, yeah, great answer. Okay, so if the stem cells respire, then they'll produce an output that can be measured. Do they respire?
If this is output in the CFU assay, by the end of the CFU assay, you've pushed all the progenitors that you originally plated quite far into maturity and differentiation along those erythroid and myeloid trajectories. So you aren't really looking at any HSPCs remaining if you were to harvest colonies out of the CFU assay. One thing that is important about harvesting out of the CFU assay, as I mentioned earlier, is if you have genetically modified your HSPCs, the clones that are contained within the colony, because they originated from a single progenitor, if you were to look for editing efficiency in the colony, you will be able to trace that back to the original progenitor and find out what your editing efficiency was in the functional HSPC fraction of your tested cells.
Excellent. All right, so interesting question here. So somebody says, as far as I understood, the internal reference standard is an internal sample that's been validated once. So this could be a sample of a cell suspension of aliquoted and frozen. So do you have to use one of these internal standards for every test? And what would happen if you ran out of the aliquots of the internal standard? Would you have to validate a new one?
Yeah, so this is a great question. And this has been relatively vague in the guidelines as well. The use of a reference standard using the relative potency approach is recommended. That would entail having an internal control run alongside all of your tests and having the results of your test samples be referred to back as a relative potency of your reference standard. Running out of them is a big concern. Unfortunately for this is a bioassay, so it's not having a reference standard that is an amount of calcium ions in a certain volume of liquid. We're looking at number of colonies that your reference standard can produce very efficiently and reproducibly. When you do run out of one reference standard, you do need to bridge between a new lot of reference standard and an old lot of reference standard. This is why we recommend trying to have a very large amount of sample that you can use as this internal sample. Could be a pool of quite a few different donors that are routinely tested. Having access to very large amounts of reference standard as well will allow you to see any trends that occur over time through tracking with control charts, see if there's been any difference with your methodology or any equipment or anything else that might go on in your workflow.
Excellent. All right, somebody else says, in a cord blood stability program, we're facing challenges to have sufficient colony counts from segments attached to the bag when compared to the main bag. And do you have any experiences in this regard, or can you share any advice?
Yeah, this is a tricky question that gets brought up fairly frequently. There, of course, you want to leave the main bag as untouched as possible, because it will eventually make its way into a patient if all goes well. So the representative nature of segments that you can remove, hopefully referring back to the main bag, would be quite important. If you're seeing a very large disconnect there, then you would need to internally validate the best correlation that you can with accessible portions to what actually comes out of the usable bag. And that might be different depending on your specific bag design, how you freeze, how you thaw, and how you do everything there.
Excellent. All right, can a colony become resistant to the methicult medium?
Resistant to the methicult medium. Not positive exactly what that would refer to. In the methicult medium in general, there's a large amount of cytokines, and these cytokines push the progenitors that you plate towards myeloid and erythroid differentiation. There is some difference with aging in terms of sensitivity to some cytokines in the HSPC fraction, but the resistance to them, I don't think is a possibility.
Excellent. All right. So why doesn't viability correlate with functionality?
Yeah, great question. So viability typically does, and it does quite well when cells are fresh or isolated out of culture. When it starts to fall apart, as I mentioned, is post-cryopreservation. These cells can typically look alive using methodologies that we use to essentially just determine the membrane integrity of the cells. But when time passes and the cells sort of realize that they're not feeling so great and start to activate apoptosis or necrosis pathways, then they start dying in a way that we can't figure out immediately when we look at their viability post-thaw, or even a short period of time afterwards. So some methodologies in place try to evaluate viability as soon as possible post-thaw, because they see that it gives them the highest apparent viability. And that might actually be very misleading, because if they were to monitor much later on, the viability would look quite a bit different. And of course, using a functional assay such as the CFU assay gives an actual insight into the main maintenance of functional cells in that thawed sample.
Excellent. Another good question here. Why are the colony counts so different for each sample?
Yeah, so that's terrific. These are individual donors, and humans, we are fairly different from one another. So there is going to be some inherent variability just person to person, and of course sample to sample. We can see this as well, especially if you use non-CD34 purified sources for plating in your CFU assay, if you're looking more at mononuclear cells. On a person-by-person basis, the mononuclear cells might contain very different frequencies of progenitors in them, and that will be reflected in the CFU assay as well, which is why we really do recommend plating multiple densities. So hopefully at least one or two of the densities that you plate get within a countable range of colonies that you see in the CFU assay. If you plate too few cells, you might get very few colonies or no colonies. And if you plate far too many cells, then you've hit a point of saturation where you can't really score your CFUs efficiently. If you plate multiple concentrations, hopefully one or more will be sort of within a nice range that you can count and get a good reflection of the frequency of different morphologies of colonies as well.
Fantastic. I think in the interest of time, we'll just have one last question here. Can we buy reference standards for the CFU assay?
Yeah, fantastic question. So reference standards need to be validated internally. So there's nothing that can be bought pre-validated. However, there are primary samples that stem cell cells that could be purchased, and if desired, pool quite a large number of these and validate those internally to have available as reference standards moving forward. Great question.
Excellent. Well, yeah, thanks so much, Colin, for all the great insights in your presentation, as well as the Q&A. It's really been a pleasure having you with us.
Great. Thank you very much, Liam. Thank you, everyone, as well, for attending today's webinar, and I hope the rest of your day goes well.
Yeah, definitely. Big thanks to the audience for participating. And in closing, we hope you enjoyed the Scientist.com and Stem Cell Technologies webinar, and we'll see you again next time. Have a great day, everyone.